Pharmakokinetics, PK, is the study of how the body interacts with administered substances for the entire duration of exposure, medications for the sake of this article. This is closely related to but distinctly different from pharmacodynamics, which examines the drug's effect on the body more closely. The four main parameters generally examined by this field include absorption, distribution, metabolism, and excretion, ADME. Wielding an understanding of these processes allows practitioners the flexibility to prescribe and administer medications that will provide the greatest benefit at the lowest risk and allow them to make adjustments as necessary, given the varied physiology and lifestyles of patients. Go to issues of concern. Absorption. Absorption is the process that brings a drug from the administration, example, tablet, capsule, into the systemic circulation. Absorption affects the speed and concentration at which a drug may arrive at its desired location of effect, example, plasma. There are many possible methods of drug administration, including but not limited to oral, intravenous, intramuscular, intrathecal, subcutaneous, buccal, rectal, vaginal, ocular, otic, inhaled, nebulized, and transdermal. Each of these methods has its own absorption characteristics, advantages, and disadvantages. The process of absorption also often includes liberation, or the process by which the drug is released from its pharmaceutical dosage form. This is especially important in the case of oral medications. For instance, an oral medication may be delayed in the throat or esophagus for hours after being taken, delaying the onset of effects or even causing mucosal damage. Once in the stomach, the low pH may begin to chemically react with these drugs before they even arrive in the systemic circulation. 1. Bioavailability. Bioavailability is the fraction of the originally administered drug that arrives in systemic circulation and depends on the properties of the substance and the mode of administration. It can be a direct reflection of medication absorption. For example, when administering medication intravenously, 100% of the drug arrives in circulation virtually instantly, giving this method a bioavailability of 100%. 2. This makes intravenous administration the gold standard regarding bioavailability. This concept is especially important in orally administered medications. Oral medications, once swallowed, must navigate the acidity of the stomach and be taken up by the digestive tract. The digestive enzymes begin the process of metabolism for oral medications, already diminishing the amount of drug arriving in circulation before being taken up. Once absorbed by gut transporters, the medications then often have to undergo first-pass metabolism. When oral medication is administered, it is often processed in large quantities by the liver, gut wall, or digestive enzymes, subsequently lowering the amount of medication that arrives in circulation, therefore, having a lower bioavailability. 2. These processes will be discussed in greater detail under metabolism. Other modes of administration may delay certain quantities of drugs to arrive in circulation at the same time, intramuscular, oral, transdermal, giving rise to the use of the area under the plasma concentration curve, AUC. The AUC is a method of calculating the drug bioavailability of substances with different dissemination characteristics, and this observes the plasma concentration over a given time. By calculating the integral of that curve, bioavailability can be expressed as a percentage of the 100% bioavailability of intravenous administration. Distribution. Distribution describes how a substance is spread throughout the body. This varies based on the biochemical properties of the drug as well as the physiology of the individual taking that medication. In its simplest sense, the distribution may be influenced by two main factors, diffusion and convection. 3. These factors may be influenced by the polarity, size, or binding abilities of the drug, the fluid status of the patient, hydration and protein concentrations, or the body habitus of the individual. 4. The goal of the distribution is to achieve what is known as the effective drug concentration. This is the concentration of the drug at its designed receptor site. To be effective, a medication must reach its designated compartmental destination, described by the volume of distribution, and not be protein-bound in order to be active. Volume of distribution, VD. This metric is a common method of describing the dissemination of a drug. It is defined as the amount of drug in the body divided by the plasma drug concentration. 4. It is important to remember that the body is made up of several theoretical fluid compartments, extracellular, intracellular, plasma, etc., and VD attempts to describe the fictitious homogeneous volume in a theoretical compartment. When a molecule is very large, charged, or primarily protein-bound in circulation, such as the GnRH antagonist cetrorelix, Vd equals 0.39 L/kg, it stays intravascular, unable to diffuse, reflected by a low Vd. A different molecule that is smaller and hydrophilic would have a larger Vd reflected by its distribution into all extracellular fluid. Finally, a small lipophilic molecule, such as chloroquine, Vd equals 140 L/kg, would have a very large Vd as it can distribute throughout cells and into adipose tissues. 5. There may be multiple volumes of distribution depending on the rate of distribution within the subject. 4. Knowledge of the volume of distribution is an important factor for a practitioner to understand dosing schemes. For example, an individual with advanced infection may require a loading dose of vancomycin to achieve desired trough concentrations. A loading dose allows the drug concentrations to rapidly achieve their ideal concentration instead of needing to accumulate before becoming effective. It is directly related to the volume of distribution and is calculated by Vd times the desired plasma concentration divided by bioavailability. 6. Protein binding. In the body, a drug may be protein-bound or free. Only free drug can act at its pharmacologically active sites, example, receptors, cross into other fluid compartments, or be eliminated. In the clinical setting, the free concentration of a drug at receptor sites in plasma more closely correlates with effect than is the total concentration in plasma. 4. The protein binding of the substance largely determines this. Any reduction in plasma protein binding increases the amount of drug available to act on receptors, possibly leading to greater effect or an increased possibility of toxicity. The principal proteins responsible for binding drugs of interest are albumin and alpha-acid glycoprotein. 7. These proteins may fluctuate depending on the age and development of the patient, any underlying liver or kidney disease, or nutrition status. One example in which this is relevant is renal failure. In renal failure, uremia decreases the ability of acidic drugs, such as diazepam, to bind to serum proteins. Even though the same amount of drug is initially given, there is far more drug in the active space, unbound by serum protein. This will increase the effect of the medication and increase the possibility of toxicity, example, respiratory depression. 4. Metabolism. Metabolism is the processing of the drug by the body into subsequent compounds. This is often used to convert the drug into more water-soluble substances that will progress to renal clearance or, in the case of prodrug administration such as codeine, metabolism may be required to convert the drug into active metabolites. 8. Different strategies of metabolism may occur in multiple areas throughout the body, such as the gastrointestinal tract, skin, plasma, kidneys, or lungs, but the majority of metabolism is through phase I, CYP450, and phase II, UGT, reactions in the liver. Phase I reactions generally transform substances into polar metabolites by oxidation, allowing conjugation reactions of phase II to take place. 2. Most commonly, these processes inactivate the drug, convert it into a more hydrophilic metabolite, and allow it to be excreted in the urine or bile. Excretion. Excretion is the process by which the drug is eliminated from the body. The kidneys most commonly conduct excretion, but for certain drugs, it may be via the lungs, skin, or gastrointestinal tract. In the kidneys, drugs may be cleared by passive filtration in the glomerulus or secretion in the tubules, complicated by reabsorption in some compounds. Clearance. Clearance is an essential term when examining excretion. It is defined as the ratio of the elimination rate of a drug to the plasma drug concentration. This is influenced by the drug, blood flow, and organ status, usually kidneys, of the patient. In the perfect extraction organ, in which blood would completely be cleared of medication, the clearance would become limited by the overall blood flow through the organ. 4. An understanding of clearance allows practitioners to calculate appropriate dosing rates of medications. Maintenance dosing ideally replaces the amount of drug that was eliminated since the previous administration. 9. It is calculated by clearance times the desired plasma concentration divided by bioavailability. Half-life, T. The half-life is the amount of time for serum drug concentrations to decrease by 50%. Defined by the equation T SAD phase 0.693 xVd/clearance, it is directly proportional to the volume of distribution and inversely to clearance. The half-life of medications often becomes altered from changes in the clearance parameters that come with disease or age. 4. 10. Drug kinetics. This is the graphical manifestation of metabolism and excretion and is the depiction of a medication's half-life. The two major forms of drug kinetics are described by zero-order versus first-order kinetics. Zero-order kinetics display a constant rate of metabolism and/or elimination independent of the concentration of a drug. This is the case with alcohol and phenytoin elimination. There is a variable half-life that decreases as the overall serum concentrations decrease. In contrast, first-order kinetics relies on the proportion of the plasma concentration of the drug. First order has a constant T with decreasing plasma clearance over time. This is the major elimination model of most medications. These two models are not usually independent for most drugs. However, as is the case with salicylates, at concentrations below 1.4 millimoles per liter, elimination is proportional to serum concentrations while, at higher concentrations, elimination is constant due to saturation of metabolic and eliminatory processes. 11. These kinetic models can be used to estimate steady states and complete elimination of medications. Steady state is when the administration of a drug and the clearance are balanced, creating a plasma concentration that is unchanged by time. Under ideal treatment circumstances, in which a drug is administered by continuous infusion, this is achieved after treatment has been operational for 4 to 5 half-lives. This is the point at which the system is said to be in a steady state. This steady-state concentration can only be altered by changes in dosing interval, total dose, or changes in the clearance of the drug. Similarly, total elimination is measurable by half-lives. Upon administration of a drug that follows first-order elimination kinetics, it may be assumed that it is completely eliminated by 4 to 5 half-lives as, by that point, 94 to 97% of the medication has left the system. For example, the T of morphine is 120 minutes. Therefore, one may assume that there is a negligible amount of morphine in a patient's system 8 to 10 hours after administration. 12. Go to clinical significance. When a provider prescribes medication, it is with the ultimate goal of a therapeutic outcome while minimizing adverse reactions. A thorough understanding of pharmakokinetics is essential in building treatment plans involving medications. Pharmakokinetics, as a field, attempts to summarize the movement of drugs throughout the body and the actions of the body on the drug. By using the above terms, theories, and equations, practitioners can better estimate the locations and concentrations of a drug in different areas of the body. The appropriate concentration needed to obtain the desired effect and the amount needed for a higher chance of adverse reactions is determined through laboratory testing. Using the equations given above, a clinician can easily estimate safe medication dosing over a period of time and how long it will take for a medication to leave a patient's system. These are, however, statistically based estimations, influenced by differences in the drug dosage form and patient pathophysiology. This is why a deep understanding of these concepts is essential in medical practice so that improvisation is possible when the clinical situation requires it. Nursing, allied health, and interprofessional team interventions. The interprofessional team members caring for the patient need to work together to ensure the safety and efficacy of pharmacotherapy. The patient may require training on how to correctly self-administer and store their medications. The physician, nurse, or pharmacist can perform this education, and in fact, it may serve the patient well to hear it from multiple providers to optimize therapy and minimize toxicity. Importantly, the interprofessional team needs to monitor for signs of drug efficacy and toxicity, which are affected by the drug's pharmacokinetic parameters, example, half-life. The pharmacist should verify the dosing, perform a drug interaction check, and follow the plasma concentrations of medication if clinically warranted, example, gentamicin. Nursing can monitor adverse events and make preliminary assessments of treatment effectiveness on subsequent visits, as well as verifying patient medication adherence. Both nurses and pharmacists need to have an open communication line with the prescribing physician so they can report or discuss any concerns regarding drug therapy or the patient's drug regimen in general. This type of interprofessional communication is necessary to optimize patient outcomes with minimal adverse events. Most drugs undergo chemical alteration by various bodily systems to create compounds that are more easily excreted from the body. These chemical alterations occur primarily in the liver and are known as biotransformations. Becoming knowledgeable about these alterations in chemical activity is crucial in utilizing the optimal pharmacological intervention for any patient and is thus of interest to any provider who routinely treats patients with medication. This activity covers drug metabolism, biotransformations, and polypharmacy and highlights the role of the interprofessional team in caring for patients on multiple medications. Objectives. Identify the types of drug biotransformation in the body. Describe the issues associated with drug induction. Review the phases of drug metabolism. Explain interprofessional team strategies for improving care coordination and communication to advance drug safety and improve outcomes. Access free multiple choice questions on this topic. Go to Introduction. Drug metabolism is a crucial aspect of medical practice and pharmacology. Most drugs undergo chemical alteration by various bodily systems to create compounds that are more easily excreted from the body. These chemical alterations occur primarily in the liver and are known as biotransformations. Understanding these alterations in chemical activity is crucial in utilizing the optimal pharmacological intervention for any patient and is thus of interest to any provider who routinely treats patients with medication. 1, 2, 3, 4. Go to Function. Biotransformations occur by mechanisms categorized as either phase I, modification, phase II, conjugation, and in some instances, phase III, additional modification and excretion. Phase I modifications alter the chemical structure of the drug, usually by oxidation, reduction, hydrolysis, cyclization/decyclization, and the removal of hydrogen or the addition of oxygen. In some instances, this process will change an inactive prodrug into a metabolically active drug. Oxidation typically results in metabolites that still retain some of their pharmacological activity. For example, the common anxiolytic drug diazepam is transformed into desmethyldiazepam and then to oxazepam by phase I modification. Both of these metabolites produce similar physiological and psychological effects of diazepam itself. Phase II modifications involve reactions that couple the drug molecule with another molecule in a process called conjugation. Conjugation usually renders the compound pharmacologically inert and water-soluble, so that the compound can easily be excreted. Mechanisms of conjugation include methylation, acetylation, sulfation, glucuronidation, and glycine or glutathione conjugation. These processes can occur in the liver, kidney, lungs, intestines, and other organ systems. An example of phase II metabolism is when oxazepam, the active metabolite of diazepam, is conjugated with a molecule called glucuronide such that it becomes physiologically inactive and is excreted without further chemical modification. 5. Following phase II metabolism, phase III may also occur, where the conjugates and metabolites can be excreted from cells. 6. A critical factor in drug metabolism is enzymatic catalysis of these phase I and phase II processes. The type and concentration of liver enzymes are crucial to the efficient metabolism of drugs. The most important enzymes for medical purposes are monoamine oxidase and cytochrome P450. These two enzymes are responsible for metabolizing dozens of biogenic and xenobiotic chemicals. Monoamine oxidase, as the name suggests, catalyzes the processing of monoamines such as serotonin and dopamine. Monoamine oxidase inhibitors, MAOI, are used as antidepressants as they increase CNS concentrations of serotonin and dopamine. Cytochrome P450 catalyzes the metabolism of many psychoactive drugs, including amphetamines and opioids. 7. Go to Issues of concern. Drug metabolism can affect the plasma concentrations of drugs. For example, prescribers need to be concerned about drug-drug interactions. For example, if rifampin is taken concomitantly with imatinib, imatinib's plasma concentrations can be reduced because rifampin can induce CYP3A4 activity. Thus, imatinib's anti-cancer activity can be attenuated. 8. Go to Clinical significance. In any kind of pharmacological intervention, it is essential to consider how and when a specific drug is eliminated from the body. Most of the time, drug clearance takes place according to first-order kinetics. In other words, the rate of clearance depends on the plasma concentration of the drug. That is, the rate of elimination is proportional to drug concentration. The rate of this form of clearance depends on the chemical in question and is often represented by half-life. This is the duration of time it takes for 50% of the drug to be eliminated from the body. For example, the half-life of cocaine is approximately 1 hour, thus, after 4 hours, only about 6.25% of the initial dose is present in the body. However, the elimination of some drugs occurs at a constant rate that is independent of plasma concentrations. Ethanol is one example. It is eliminated at a constant rate of about 15 milliliters per hour regardless of the concentration in the bloodstream. This is called zero-order kinetics and occurs when enzyme binding sites are saturated at low concentrations. Kinetics are of interest in medicine because monitoring of drug concentration is often of clinical importance with many medications. An understanding of pharmakokinetics, specifically drug elimination, allows providers to alter therapies in a patient-specific fashion. The goal of therapy is to achieve a steady-state plasma concentration at which drug metabolism and elimination occur at equal rates. Metabolism is a highly variable process that can be influenced by a number of factors. One major disruptor of drug metabolism is depot binding, meaning the coupling of drug molecules with inactive sites in the body, such that the drug is not accessible for metabolism. This action can affect the duration of action of pharmacological agents susceptible to depot binding. One notable example is tetrahydrocannabinol, THC, the main psychoactive component of marijuana. THC is highly lipid-soluble and depot binds in the adipose tissue of users. This interaction drastically slows the metabolism of the drug, which is why metabolites of THC can be detected in urine weeks after the last use, 9-10. Another factor in drug metabolism is enzyme induction. Enzymes are induced by repeated use of the same chemical. The body becomes accustomed to the constant presence of the drug in question and compensates by increasing the production of the enzyme necessary for the drug's metabolism. This contributes to pharmacological tolerance and is one reason why patients need ever-increasing doses of certain drugs to produce the same effect. Opioids are a prime example. Patients with long-term prescriptions for opioid analgesics will notice that their medication becomes less effective over time. Notably, induction will increase the metabolic rate for all drugs processed via the enzyme induced. For example, chronic amphetamine use will induce higher concentrations of the enzyme CYP2D6. This enzyme is also important in the metabolism of certain opioids, such as oxycodone. Thus, a physician prescribing oxycodone to a patient using amphetamines would have to give the patient a higher dose to produce the desired effect. 11. In contrast, some drugs have an inhibitory effect on enzymes, making the patient more sensitive to other medications metabolized through the action of those enzymes. A classic example is the inhibition of monoamine oxidase by certain antidepressant drugs. These compounds produce their psychotherapeutic effects by blocking the enzyme that breaks down pleasure chemicals in the brain. However, this can cause problems when patients on an MAOI take drugs that cause abnormally high concentrations of these neurochemicals. A patient on an MAOI who uses cocaine, which elevates the concentration of serotonin, dopamine, and norepinephrine, will experience a much more potent effect from the cocaine. This interaction can lead to numerous physiological problems, including tachycardia, hypertension, and serotonin syndrome. 12. Drugs that share elements of their metabolic pathways can also compete for the same binding sites on enzymes, decreasing the efficiency of their metabolism. For instance, alcohol and certain sedatives are metabolized by the same member of the cytochrome P450 family. Only a limited number of enzymes exist to break these chemicals down. Thus, if a patient were administered pentobarbital while also metabolizing alcohol, the pentobarbital would not be completely metabolized because most of the necessary enzymes would be filled by alcohol molecules. This is one reason that alcohol and other sedative/hypnotic drugs can have a synergistic effect when co-administered. Go to other issues. Depending on whether a drug is metabolized and eliminated renally or hepatically, impairment in either of these systems can significantly alter dosing, dose intervals, therapeutic effect, and even whether a particular drug can be used at all. Go to enhancing healthcare team outcomes. Drug metabolism is a very important clinical concern for the interprofessional healthcare team. Clinicians, nurses, and especially pharmacists need to work together to prevent clinically significant drug interactions that could affect patients' health. In a hospital setting, nurses must be alert for signs of a toxic buildup of metabolites or active drugs, particularly in instances of renal or hepatic insufficiency, so that they can alert the clinician and pharmacist. In many cases, drugs such as aminoglycoside antibiotics, warfarin, fluoroquinolones, etc., are dosed and monitored by pharmacists, who monitor serum levels of the drugs and renal function. This points to the importance of an interprofessional approach to drug dosing and administration in light of the effects of drug metabolism on patients, whether through impaired metabolism, drug-drug interactions, enzymatic induction, or other factors. Using the interprofessional paradigm will result in better therapeutic results with fewer adverse events. A drug interaction is a reaction between two or more drugs or between a drug and a food, beverage, or supplement. Taking a drug while having certain medical conditions can also cause a drug interaction. For example, taking a nasal decongestant if you have high blood pressure may cause an unwanted reaction. A drug interaction can affect how a drug works or cause unwanted side effects. Treatment with HIV medicines, called antiretroviral therapy or ART, helps people with HIV live longer, healthier lives and reduces the risk of HIV transmission. But drug interactions can complicate HIV treatment. Healthcare providers carefully consider potential drug interactions before recommending an HIV treatment regimen. Before taking HIV medicines, tell your healthcare provider about all prescription and non-prescription medicines, vitamins, nutritional supplements, and herbal products you are taking or plan to take. What is a drug interaction? Medicines help us feel better and stay healthy. But sometimes drug interactions can cause problems. There are 3 types of drug interactions. Drug-drug interaction, a reaction between two or more drugs. Drug-food interaction, a reaction between a drug and a food or beverage. Drug-condition interaction, a reaction that occurs when taking a drug while having a certain medical condition. For example, taking a nasal decongestant if you have high blood pressure may cause an unwanted reaction. A drug interaction can affect how a drug works or cause unwanted side effects. Do HIV medicines ever cause drug interactions? Treatment with HIV medicines, called antiretroviral therapy or ART, helps people with HIV live longer, healthier lives and reduces the risk of HIV transmission. But drug interactions, especially drug-drug interactions, can complicate HIV treatment. Drug-drug interactions between different HIV medicines and between HIV medicines and other medicines are common. Before recommending an HIV treatment regimen, healthcare providers carefully consider potential drug-drug interactions between HIV medicines. They also ask about other medicines a person may be taking. For example, some HIV medicines may make hormonal birth control less effective, so women using hormonal contraceptives may need to use an additional or different method of birth control to prevent pregnancy. For more information about using birth control and HIV medicines at the same time, view the HIV and birth control infographic from HIVinfo. Can drug-food interactions and drug-condition interactions affect people taking HIV medicines? Yes, the use of HIV medicines can lead to both drug-food interactions and drug-condition interactions. Food can affect the absorption of some HIV medicines and increase or reduce the concentration of the medicine in the blood. Depending on the HIV medicine, the change in concentration may be helpful or harmful. Directions on how to take HIV medicines specify whether to take the medicine with food or on an empty stomach. Some HIV medicines can be taken with or without food, because food does not affect their absorption. Conditions, such as kidney disease, hepatitis, and pregnancy, can affect how the body processes HIV medicines. The dosing of some HIV medicines may need to be adjusted in people with certain medical conditions. How can a person avoid drug interactions? You can take the following steps to avoid drug interactions. Tell your healthcare provider about all prescription and non-prescription medicines you are taking or plan to take. Also tell your healthcare provider about any vitamins, nutritional supplements, and herbal products you take. Tell your healthcare provider about any other conditions you may have, such as high blood pressure or diabetes. Before taking a medicine, ask your healthcare provider or pharmacist the following questions. What is the medicine used for? How should I take the medicine? While taking the medicine, should I avoid any other medicines or certain foods or beverages? Can I take this medicine safely with the other medicines that I am taking? Are there any possible drug interactions I should know about? What are the signs of those drug interactions? In the case of a drug interaction, what should I do? Take medicines according to your healthcare provider's instructions. Always read the information and directions that come with a medicine. Drug labels and package inserts include important information about possible drug interactions. Drug treatment is in many cases ineffective. Besides, patients who do not respond to the treatment can develop so-called adverse drug reactions, ADRs, as a consequence of the treatment, cause over 100,000 deaths per year in USA. Pharmakogenetics is the discipline which takes the patient's genetic information of drug transporters, drug metabolizing enzymes and drug receptors into account, leading to optimal choice and dose of the drugs in question. It represents a kind of patient-oriented medicine or personalized medicine. It is believed that a lot of costs for the society can be saved by so-called individualized drug therapy, mainly based on pharmacogenetic data. Genetic variations, mostly single nucleotide polymorphisms, SNPs and microsatellites, can be present in drug transporters, in majority of phase I and phase II drug metabolizing enzymes or in drug receptors. SNPs in drug transporters could alter the absorption, distribution and the elimination of the drug from the body. The polymorphisms in drug metabolizing enzymes can cause abolished, quantitatively or qualitatively altered or enhanced drug metabolism. Several examples exist where subjects carrying certain alleles suffer from a lack of drug efficacy because of ultra-rapid metabolism caused by multiple genes or by induction of gene expression. On opposite site, adverse effects can develop in slow metabolizers as a result of the accumulation of drug in presence of defective alleles. Finally, certain polymorph genes can be used as markers for optimization of the drug therapy. It is likely that predictive genotyping is of benefit in 10 to 20% of drug treatment and thereby allows for prevention the appearance of ADRs and thus improves the health for this part of the patients. Go to 7.2 Drug receptors. Receptor is a macromolecule in the membrane or inside the cell that specifically, chemically, bind a ligand, drug. The binding of a drug to receptor depends on types of chemical bonds that can be established between drug and receptor. The strength of this chemical bonds, covalent, ionic, hydrogen, hydrophobic, determine the degree of affinity of ligand to receptor. Ligands, drugs, that attracted the receptors may be classified as agonists or antagonists. Agonists produce the biological response as a result of receptor-ligand interactions, therefore agonists possess efficacy. On the contrary, antagonists did not provoke any biological activity after binding to its receptor. There are different types of receptors. 1. Transmembrane ion channels receptors. Transmembrane G-protein coupled receptors. Transmembrane receptors with cytosolic domain. Intracellular, cytoplasm or nucleus, receptors. 7.2.1 Transmembrane ion channels receptors. The most rapid cellular responses to receptor activation are mediated via ligand-gated ion channels. These kind of transmembrane receptors composed of multiple peptide subunits and each of it contains 4 membrane-spanning domains. The ligand binding causes the conformational changes of receptor and ion channel forming. The binding of ACH to each of 4 subunits of AChR induces change in receptor and opening the sodium-selective channel through the center of the receptor protein. It causes the depolarization of surrounding membrane. In this type of receptors belong nicotinic acetylcholine receptors and receptors for GABA, serotonin and some other neurotransmitters. 7.2.2 Transmembrane G-protein coupled receptors. The most abundant type of drug receptors are G-protein coupled receptors, GPCR. This are family of, over 100 different, transmembrane receptors which share a well-conserved structure and transduce their signals via activation of intracellular guanidine nucleotide binding protein, G-protein, figure 7.2. A variety of ligands for this receptors include biogenic amines, ACH, noradrenaline, serotonin, amino acid neurotransmitters, glutamate, glycine, and peptide hormones, angiotensin II, somatostatin. There are multiple GPCR types for a single ligand. The result is the possibility that single ligand can activate a variety of transduction pathways. Thus receptor is defined not only just by which ligands binds to it but also by second messenger systems, CAMP, PLC, Na/H exchange, and signal transduction pathway, which is activated by receptor activation. Transmembrane receptors with cytosolic domain. The intracellular domain of this transmembrane receptor is either enzymatic active, catalytic receptors, or is bound to specific enzymes in cytosol, enzyme-coupled receptors. The catalytic receptors are activated predominantly by peptide hormones, insulin, growth factors, etc. Catalytic part of receptors has the protein kinase activity. Mostly dimerization of catalytic as well as enzyme-coupled receptors is necessary for kinase activity. Phosphorylation of intracellular proteins by these receptors results in effects such as opening the ion channels, initiation of gene expression or as in the case of enzyme-coupled receptors activation of signal transducers and activators like JAKs and STATs. 7.2.4 Intracellular, cytoplasm or nucleus, receptors. Those receptors are not associated with cell membrane. In general, their protein molecule consists from three main domains, HSB90 and DNA and ligand binding domains. Ligands are mostly lipid-soluble and passively pass cell membrane. Agonists include nitric oxide, steroid hormones and vitamin D. Ligand binding activates receptor and initiates the dissociation from HSB90. The complex then translocates to nucleus and bind to specific DNA sequences mostly located in gene promoter region, figure 7.3. This kind of signal transduction is slow, but duration of response can last long. Pharmakokinetics of human beta-adrenergic receptors. Beta-adrenergic receptors, ADRBs, are transmembrane G-protein-coupled receptors that bind adrenaline or noradrenaline in sympathetic nervous system. There are three types of ADRBs, ADRB1, ADRB2, and ADRB3. ADRB3 has been least studied to date, and the role of ADRB3 in cardiovascular disease is not known. ADRB1 are the predominant type expressed in the heart. ADRB2 are abundantly expressed in bronchial smooth cells, and activation of them results in bronchodilation. ADRB1 and ADRB2 are intronless genes encoding 477 and 413 amino acid proteins, respectively. They share a common structure with an extracellular amino terminus domain, 7 transmembrane spanning domains, and a cytoplasmic carboxyl terminus. Binding of ligand, adrenaline or noradrenaline or other agonists, to these receptors coupled to G protein lead to conversion of ADP to CAMP. Increased CAMP stimulates a chain of events that culminates with removal of calcium from contractile protein and increase the activation of contraction through greater calcium cycling. The global effect is improved systolic and diastolic function, too. 7.3.1 ADRB1 polymorphisms. 23 polymorphisms have been described and 13 of these change amino acid sequence. As ADRB1 has not a recognized role in asthma, the majority of association and pharmacogenetic studies were done by cardiovascular phenotypes. SER49-GLY and ARG389-GLY have been widely studied owing to studies supporting the functional effects on ADRB1 activity and phenotypes. The GLY49 allele showed altered glycosylation and a more pronounced agonist-induced receptor down-regulation in a fibroblast model. What could explain the resistance to chronic beta-adrenergic stimulation through diseases or medication? The ARG489 alleles showing higher basal levels of CAMP. Thus, the ARG389 may have inherently coupling to GS and increase the signal transduction. Population association studies show SER49 homozygotes have higher mean heart rates and are associated with reduced 5-year survival. However, different studies gave divergent results. The ARG389 variant is associated with elevated diastolic blood pressure and higher resting heart rate. This finding was strengthened by similar conclusions of different studies. Furthermore, ARG389-GLY polymorphism appears to interact with 4-amino acid deletion in the alpha-2 adrenergic receptor gene. Homozygotes with both polymorphisms have increased risk for heart failure, odd was 10:1. However, it is approved in Black population. The number of double homozygotes in Caucasians was too small, 2. ADRB2 polymorphisms. From 12 SNP identified, only 5 of these predict the change of amino acid sequence. The most studied SNPs are ARG16-GLY, GLN27-GLY, and THR164-IL. Functional analyses showed that the first 2 SNPs downregulate ADRB2 in fibroblasts and enhanced isoproterenol-mediated desensitization in humans carrying the ARG16. The IL-164 allele has increased binding affinity for endogenous ligands. SNPs in ADRB2 have been studied in patients with asthma, obesity, and diabetes. Recent studies concluded that GLY16 alleles predisposes to nocturnal asthma and asthma severity as well as to response to beta-agonist therapy, albuterol, in asthmatics. Association studies of ADRB2 gene variations with hypertensive phenotype yielded mixed results. In healthy individuals, ADRB1 is far more abundant than ADRB2. However, in the heart failure diseases, proportion of ADRB2 is increased to 40%. This data could be interpreted to mean that ADRB2 SNPs might have great effects on heart failure. 7.3.3 Pharmacogenetic studies of ADRBs. As the ADRBs are the major target for pharmacological therapy, beta receptor agonists and antagonists, some of pharmacogenetic studies were published. Studies showed that ARG389 homozygotes have increased effects of ADRB1 agonist dobutamine and ADRB1 antagonist metoprolol as compared to GLY389 homozygotes. SNPs in ADRB2 influence the response to methacholine and albuterol therapy of asthma. In conclusion, for ADRB1, the GLY49 and ARG389 alleles seem to be variants that perform more favorably in heart failure patients given beta blockers. In case of ADRB2, the data suggest a negative effect of ARG16 alleles on short-term beta agonist therapy. 2. Go to 7.4 Pharmakokinetics of estrogen receptors. 7.4.1 Type and function of estrogen receptors. Estrogen receptors, ERs, are intracellular nuclear receptors that belong to steroid hormone receptor family. There are two types of ER alpha, ER1, and ER beta, ER2, encoded by two different genes ESR1 and ESR2, respectively. ESR1 and ESR2 are expressed mostly bones, breasts, ovaries, cardiovascular system and central neural system, but ESR2 mRNA was also found in kidney, lung, colon and testis tissues. ERs function as transcription factors activated by a ligand. Ligands bind to ERs are endogenous estrogen hormones, estradiol, or estrogens administered at hormone replacement therapy, HRT. A new group of drugs, selective estrogen receptor modulators, SERMs, act as ER agonist in a specific tissue, like raloxifene in bone, but as an ER antagonist in other tissue, like tamoxifen in breast. Irrespective of ligand binding to ER results in activation and translocation of ERs to nucleus, where the complexes bind to specific DNA sequences, estrogen-responsive element. In association with other coactivators and repressors alters the expression of target genes. For example, binding of ER1 ligand complex to Phos/Jun complex facilitate the binding of Phos/Jun heterodimer to AP1 site and activation of many genes including IGF-1. On the contrary, ESRs inhibit binding of NF-κB to IL-6 promoter. 7.4.2 Pharmakokinetics of ERs. Pharmakokinetics of ERs was mostly studied regarding the role of ERs genes polymorphisms in determine the response to estrogen and SERM therapy and the risk of cardiovascular events. Clinical data of hormone replacement therapy, HRT, for maintain the health of postmenopausal women showed an increase in cardiovascular complications as the adverse drug reactions. However, our data at raloxifene, SERM, treatment showed opposite effect with lowering of cholesterol level. Both estrogens and SERM's activity could be modified with genetic polymorphisms of ER alpha gene. In this regard, 10 different SNPs or microsatellite polymorphisms over regulatory, coding and uncoding regions were analyzed. Cluster of 4 SNPs located at the 3' end of intron 1 were significantly associated with higher increase of HDL level at HRT treatment. Among these SNPs, the IVS-1-401T greater than C was most strongly related to HDL increase. 3. Our data prove positive effects of exonic but not intronic SNPs on total cholesterol level at postmenopausal osteoporotic women treated with raloxifene. 4. Several other studies have also examined the effects of ER1 IVS-1-401T greater than C polymorphism on response to HRT or bisphosphonates treatment of osteoporosis. IVS-1-401C allele was associated with greater effects of estrogen on bone mineral density. Our data on nearly 60 postmenopausal women treated with raloxifene didn't show any influence of ER1 SNPs on SERM effect. Conclusion. The information about the role of polymorph drug receptors for efficiency of drug therapy are more scarce, although promising examples are seen in drug treatment of asthma where the efficiency can be severely enhanced by predictive genotyping of the drug targets. To move the field to clinical practice, future studies should be larger and have to consider the complexity of drug response, receptor protein with proteins in signal transduction. Finally, for those drugs with multiple pharmacological effects or effects in different organs, the genetic contributions have to be considered separately. 5. Discovering highly predictive genotype-phenotype associations during drug development and demonstrating their clinical validity and utility in well-designed prospective clinical trials will no doubt better define the role of pharmacogenetics in future clinical practice. What is pharmacovigilance? Medicines and vaccines have transformed the prevention and treatment of diseases. In addition to their benefits, medicinal products may also have side effects, some of which may be undesirable and/or unexpected. Pharmacovigilance is the science and activities relating to the detection, assessment, understanding and prevention of adverse effects or any other medicine/vaccine-related problem. All medicines and vaccines undergo rigorous testing for safety and efficacy through clinical trials before they are authorized for use. However, the clinical trial process involves studying these products in a relatively small number of selected individuals for a short period of time. Certain side effects may only emerge once these products have been used by a heterogeneous population, including people with other concurrent diseases, and over a long period of time. First Joint Meeting of WHO GACS and WHO ACISO-MP. The report from the first joint meeting of WHO Global Advisory Committee on Vaccine Safety and WHO Advisory Committee on Safety of Medicinal Products is now available. An integrated WHO pharmacovigilance team was established in 2020 to combine work related to the safety of medicines and vaccines within the Department of Regulation and Prequalification, RPQ. Following this transformation, WHO convened a joint meeting with both the Advisory Committee on Safety of Medicinal Products, ACISO-MP, and the Global Advisory Committee on Vaccine Safety, GACVS, for the first time on 14 to 16 June 2022. Second Joint Meeting of WHO GACS and WHO ACISO-MP. The conclusions from the second joint meeting of the WHO GACS and the WHO ACISO-MP, 14 to 16 December 2022, are now available, Weekly Epidemiological Report and ACISO-MP recommendations. The meeting discussed various topics including pox, monkeypox, vaccines and safety surveillance, safety monitoring of pregnant and breastfeeding women, active safety surveillance of molnupiravir, hallucinations reported with delamanid in children, dolutegravir exposure at conception, emergency use protocol for tecovirimat, the use of sodium valproate in women and girls of childbearing potential as well as a gaming approach to risk and safety communication. Results of literature review and survey of COVID-19 vaccine safety assessment activities. In 2021, there was an urgent need to support the COVAX Vaccine Safety Working Group, VSWG, the Coalition for Epidemic Preparedness Innovations, CEPI, and the WHO Pharmacovigilance Team, WHOPV, in collating the studies evaluating the safety of COVID-19 vaccines globally to inform strategy and policy. A landscape assessment of COVID-19 vaccine safety surveillance activities, including a literature review and a survey, was conducted to inform on potential sources of evidence and to identify gaps, VH safety maps. Large language models, LLMs, such as ChatGPT promise advances that extend beyond capturing public interest. Because transformer models like GPT have an architecture that supports the understanding of language in context, they point to an array of novel possibilities for scientific research. The transformer architecture is critical, according to Michael Connell, the Chief Operating Officer at InThought. In a recent interview, Connell provided a sense of what to expect from generative AI in drug discovery, touching on how these tools could automate mundane tasks, streamline complex scientific workflows and speed drug discovery. The promises and pitfalls of generative AI in drug discovery. In scientific research, generative AI, of which LLMs are an example, can partly automate tasks such as summarizing academic papers, solving math problems, coding, ensuring quality compliance and annotating molecules and proteins. But the journey toward tapping the potential of LLMs in scientific research has not been without obstacles. One high-profile hiccup to apply an LLM to scientific research was Galactica, a model developed by Meta, formerly Facebook, which was yanked 3 days after introduction. A glaring shortcoming was its inability to consistently distinguish fact from fiction. In a similar vein, ChatGPT is also prone to hallucinate at times. And Nature reported that ChatGPT-written articles fool scientists. Michael Connell. Yet generative AI has been making inroads in drug discovery for the past decade. Biotech firms using an AI-first approach had more than 150 small molecule drugs in discovery and more than 15 were in clinical trials in 2022. The AI-fueled pipeline has been expanding at an annual clip of almost 40%, according to BCG. Meanwhile, drug discovery upstarts like In Silico Medicine and Atom Bioworks are deploying ChatGPT as an interface for target discovery platforms. LLM applications such as Google DeepMind's MedPA-LM show promise in answering medical queries. Additionally, NVIDIA has expanded its Clara discovery platform, a GPU-backed computational drug discovery platform that fuses AI, data analytics, simulation, and visualization to support cross-disciplinary workflows in drug design. It can handle an array of biological data formats, chemistry, protein sequences as well as DNA and RNA sequences. NVIDIA has also introduced BioNAMO, a cloud-based supercomputing framework focusing on the training and deployment of large biomolecular language models. Raising the efficiency floor. Modern Medical Research Laboratory. Scientific Lab, Drug Engineering Center full of high-tech equipment, computer screen showing DNA concept, technology for vaccine development. Modern Medical Research Lab, Gorodenkopf/Adode stock. Automating iterative loops, drug discovery has something like a knowledge-intensive, iterative marathon backed by calculated gambles. While not a silver bullet, generative AI offers the potential to automate and optimize some of the iterations in process, pointing toward more efficient pathways for discovering new drugs. As Connell suggested, if we can streamline the drug discovery process with AI, then that lowers the bar to doing good science. He continued, this kind of AI could jump to the answer, and you kind of skip a lot of the iterative work in between. Navigating complex datasets, generative AI's capacity to handle unstructured scientific data could be a game changer as well. It can significantly simplify tasks for scientists, improving both the speed and quality of analysis. I think that AI has the potential to greatly increase the efficiency of the drug discovery process, Connell said. In addition, generalist medical AI, as Nature highlighted, could potentially help interpret diverse and large sets of medical data. The company Aria Pharmaceuticals has developed an AI-based platform to find promising drug candidates. Its technology considers genetic, proteomic and metabolomic data to predict the efficacy of drug candidates. The company credits the approach in the discovery of a promising candidate for the treatment of rheumatoid arthritis, which is now in preclinical development. Reducing human errors and variability. The human-centric nature of traditional drug discovery processes may lead to variability and errors. AI has the potential to provide a consistent and precise approach, reducing these issues and resulting in a more robust drug discovery process. Owing to its iterative structure, a failure at any stage often necessitates returning to a previous stage or even starting from scratch. As Connell explains, the integration of AI in drug discovery has the potential to significantly minimize such setbacks, allowing for a more efficient process and reduced costs. Considering how generative AI would affect the efficiency of drug discovery, Connell said the trend is going to raise the efficiency baseline, the floor. Bridging knowledge gaps. The expertise required in fields like chemistry and data science can be overwhelming for individual scientists to master. By incorporating AI into research tools, they become more accessible to scientists, reducing the barriers to entry and improving efficiency. Scientists could do more if they could actually have access to scalable, compute and advanced algorithms, but it's too much for most scientists to know how to do that as well as their science, Connell said. He further emphasized the significant potential loss when chemists and data scientists work in isolation. Instead, he advocates for a more integrated approach. By placing AI tools directly in the hands of scientists, they can conceive an idea, test it, and once validated, advance it along the pipeline. Enhancing decision-making. By tapping AI in the drug discovery process, researchers could access a broader solution space, improving the decision-making process about which problems to tackle and how to approach them. Data science is also carving out a growing role in experimental chemistry, as Nature has noted. These trends could help provide a clearer path through the drug development pipeline. Reducing friction. Connell draws attention to the traditionally human-centric and sequential nature of drug discovery processes. Each step often relies on a handoff, where one expert passes their findings to the next through a report. This introduces significant friction and variability, especially when junior members are involved, potentially jeopardizing the project's success. Connell emphasized how AI can help alleviate these issues by bridging knowledge gaps, stating, AI can level that knowledge. The weakest link phenomenon becomes less a factor, you have a lot fewer weak links. He further explains how AI can automate tasks and navigate complex, unstructured data, effectively streamlining the process. I can express things in natural language and I can get technical things done behind the scenes, like querying my unstructured, complex chemical data. That's not something that's easy to create a database for. Scalability and enhanced collaboration. the capability of AI to handle vast amounts of data and rapidly scale up processes presents a major opportunity in drug discovery. As Connell put it, you want to put this power into the hands of the scientist who's got the ideas and knows how to validate them. Collaboration is an integral part of scientific discovery, and AI can bolster this aspect by promoting interdisciplinary collaboration. One of the things we work on is re-engineering the workflow or the ecosystem, Connell noted. It's almost about reshaping the people, the process, the technology, the data. How generative AI in drug discovery can raise the innovation ceiling. Augmented innovation and creativity, emphasizing the elastic capabilities of AI, Connell points out that researchers are often inspired to explore new ideas, particularly when they observe something unique in their data. Connell envisions that AI could free scientists from mundane tasks, allowing them to concentrate more on creative, innovative aspects of their work. By automating some of the more labor-intensive tasks, AI can free up scientists to focus on the creative and innovative aspects of their work. This has the potential to lead to more novel and groundbreaking discoveries in the field. Additionally, as transformer-based models advance, the acceleration of this process may occur thanks to the emergent properties they display when trained on extensive and diverse data sources, such as biomedical literature, genomic data, and chemical structures. Learning from AlphaFold and beyond, though not technically an example of generative AI, AlphaFold, a deep learning system that predicts protein structures with high accuracy, exemplifies how AI can enhance efficiency in drug discovery. By offering rapid access to protein structure information, it addresses one of the most challenging and time-consuming steps in the drug discovery process, especially for biologics. Knowledge of protein structures, however, is also prized in traditional small molecule drug discovery. Aside from AlphaFold, various AI approaches, especially machine learning algorithms such as random forest, support vector machine, neural network, deep learning and other techniques have successfully predicted absorption, distribution, metabolism, excretion and toxicity, ADME tox, properties during early drug discovery phases. Enhancing decision-making, speaking on the expansive capabilities of AI, Connell says, and if it fails, at any point, you may have to go back to the previous loop all the way back to the beginning. And so that's where the 15 years of expense comes in. Future readiness and adaptability. Embracing generative AI requires organizations to be prepared for digital transformation. This theme encapsulates the necessity for future-proofing and the adaptability for multimodal applications of generative AI. The importance of being future-proof or digitally ready is escalating, Connell said. Life science firms making strategic pivots with emergent technologies like generative AI are those that are data-ready since this area is notably data-intensive, he concluded. The pharmaceutical industry is facing many challenges to ensure drug products are free from nitrosamine contaminants and safe for human use. Since the discovery of potentially carcinogenic nitrosamines in some commonly prescribed drug products in 2018, mitigating nitrosamine risk has been a top priority for the pharmaceutical industry and its governing bodies. As such, strict regulations have been implemented by the Food and Drug Administration, FDA, and European Medicines Agency, EMA, to assess and minimize the risk of nitrosamine impurities in existing and new drug formulations. However, navigating the new legislation and choosing a suitable mitigation strategy is a complex process. Navigating nitrosamine risk mitigation with confidence. Many research groups are investigating possible root causes of nitrosamine contamination in human drug products and potential mitigation strategies. The latest data includes a large in silico study that demonstrates the risk of contamination in drug products may be more common than previously thought, with 40.4% of active pharmaceutical ingredients, API, and 29.6% of API impurities presenting as potential nitrosamine precursors. Similarly, another research group found a new impurity known as NTTP, nitroso-STG19, which was detected above the acceptable intake limit in sitagliptin, antidiabetic, medicines. What's more, scientists have uncovered an additional root cause of nitrosamine impurities, beyond the API. The nitrocellulose primer in lidding foil can act as a nitrosating agent, and nitrosamines can transfer directly from the foil to the drug product during packaging. In addition, other nitrosating agents or nitrosamine precursors like nitrites have been found in excipients. Such emerging science, regulatory changes, and additional documentation requirements can make it challenging for drug formulators and manufacturers to keep pace and develop optimized nitrosamine mitigation strategies. Partnering for success. An effective nitrosamine mitigation strategy requires robust formulation, analytical, technical, and regulatory expertise. However, many pharmaceutical companies may lack the infrastructure or experience to tackle nitrosamine risk mitigation alone. Therefore, a partnership can be the best time and cost-efficient solution to bring safer formulations to market. Whether you require help throughout your nitrosamine mitigation journey, or specialized support for certain steps, the right partner can provide you with customized solutions to succeed in this complex landscape. So what should you look for in a partner? Superior understanding of nitrosamine mitigation strategies. All companies must assess the risk of nitrosamine contamination in their drug products, as failure to do so could result in drug recalls, and potentially increase the risk of patients not receiving critical medication. If help is needed to assess nitrosamine risk, it is important to choose a partner that deeply understands the mitigation strategy you are aiming to implement. Advanced formulation and technical expertise. Adapting the manufacturing process to formulate, or reformulate, new or existing drugs, requires extensive expertise. This is not something that can be learned overnight, and therefore partnering with an expert that already has the knowledge and capabilities to assist your company is a valuable strategy. For instance, DS-Firmenich has the advanced formulation expertise to help manufacturers find an effective mitigation strategy according to their individual portfolio. Established mitigation strategy. A partner offering a proven mitigation strategy means there is no need to reinvent the wheel when it comes to creating safer drug products. Various routes of nitrosamine risk mitigation exist, such as using antioxidants as excipients, which is a valuable and safe option to lower nitrosamine formation. Results from important studies have demonstrated that the antioxidants ascorbic acid, vitamin C, and alpha-tocopherol, vitamin E, can be leveraged to successfully mitigate nitrosamine formation. An added benefit of this strategy is that the antioxidants help stabilize and protect the API from degradation. Formulators looking to use antioxidants to lower nitrosamines in drug products to below the accepted level require access to high-quality, pharma-grade, GMP-certified antioxidant ingredients. With over 70 years of experience in manufacturing the secure supply of vitamins as APIs and excipients, DS-Firmenich has the right solution for your antioxidant needs. Beyond ingredients, DS-Firmenich can also support with pioneering solutions for reformulation, based on proprietary research. Global regulatory insight. A global issue needs a global partner. To navigate the requirements of multiple regulatory agencies in various countries, as well as evolving legislations around nitrosamines, partnering with a worldwide company that can manage the necessary documentations and market entry registration processes helps companies enter the market with confidence. Finding a reliable partner that can provide the technical guidance required to reformulate existing products or develop new formulations is key to the successful implementation of a proven nitrosamine risk mitigation strategy. Anne-Cécile Bain is the Global Science and Innovation Lead, Pharma and Medical Nutrition at DS-Firmenich. Herbal medicine, a growing field with a long tradition. Traditional medicine is the knowledge, skills and practices based on the theories, beliefs and experiences indigenous to different cultures, used in the maintenance of health and in the prevention, diagnosis, improvement or treatment of physical and mental illness. World Health Organization, http://www.who.int/topic/traditional_medicine/n. There are many different systems of traditional medicine, and the philosophy and practices of each are influenced by the prevailing conditions, environment, and geographic area within which it first evolved. WHO 2005. However, a common philosophy is a holistic approach to life, equilibrium of the mind, body, and the environment, and an emphasis on health rather than on disease. Generally, the focus is on the overall condition of the individual, rather than on the particular ailment or disease from which the patient is suffering, and the use of herbs is a core part of all systems of traditional medicine. Engebretson 2002, Conboy et al. 2007, Richten 2008, Schmidt et al. 2008. Traditional Chinese medicine, TCM, is an important example of how ancient and accumulated knowledge is applied in a holistic approach in present-day healthcare. TCM has a history of more than 3,000 years. Shutian, Zhang, and Louise 2009. The book The Divine Farmer's Classic of Herbalism was compiled about 2,000 years ago in China and is the oldest known herbal text in the world, though the accumulated and methodically collected information on herbs has been developed into various herbal pharmacopoeias and many monographs on individual herbs exist. Diagnosis and treatment are based on a holistic view of the patient and the patient's symptoms, expressed in terms of the balance of yin and yang. Yin represents the earth, cold, and femininity, whereas yang represents the sky, heat, and masculinity. The actions of yin and yang influence the interactions of the 5 elements composing the universe: metal, wood, water, fire, and earth. TCM practitioners seek to control the yin and yang levels through 12 meridians, which bring and channel energy, qi, through the body. TCM is a growing practice around the world and is used for promoting health as well as for preventing and curing diseases. TCM encompasses a range of practices, but herbal medicine is a core part. Engebretson 2002, Nestler 2002, Schmidt et al. 2008, Shutian, Zhang, and Louise 2009. 3 of the top-selling botanical products, namely ginkgo biloba, allium sativum, garlic, and Panax ginseng, can be traced back to origins in TCM and are today used to treat various diseases. Li, Jiang, and Chen 2008, Shutian, Zhang, and Louise 2009. Over the past 100 years, the development and mass production of chemically synthesized drugs have revolutionized health care in most parts of the world. However, large sections of the population in developing countries still rely on traditional practitioners and herbal medicines for their primary care. In Africa, up to 90% and in India 70% of the population depend on traditional medicine to help meet their health care needs. In China, traditional medicine accounts for around 40% of all health care delivered, and more than 90% of general hospitals in China have units for traditional medicine, WHO 2005. However, use of traditional medicine is not limited to developing countries, and during the past 2 decades public interest in natural therapies has increased greatly in industrialized countries, with expanding use of ethnobotanicals. In the United States, in 2007, about 38% of adults and 12% of children were using some form of traditional medicine. Ernst, Schmidt, and Weider 2005, Barnes, Bloom, and Nahin 2008. According to a survey by the National Center for Complementary and Alternative Medicine, Barnes, Bloom, and Nahin 2008, herbal therapy or the usage of natural products other than vitamins and minerals was the most commonly used alternative medicine, 18.9%, when all use of prayer was excluded. A survey conducted in Hong Kong in 2003 reported that 40% of the subjects surveyed showed marked faith in TCM compared with Western medicine, Chan et al. 2003. In a survey of 21,923 adults in the United States, 12.8% took at least one herbal supplement, Harrison et al. 2004, and in another survey, Cato et al. 2008, 42% of respondents used dietary or nutritional supplements, with multivitamins and minerals most commonly used, followed by saw palmetto, flax, garlic, and ginkgo. At the time of the interview, the most common reasons for using traditional medicine are that it is more affordable, more closely corresponds to the patient's ideology, allays concerns about the adverse effects of chemical, synthetic medicines, satisfies a desire for more personalized health care, and allows greater public access to health information. The major use of herbal medicines is for health promotion and therapy for chronic, as opposed to life-threatening, conditions. However, usage of traditional remedies increases when conventional medicine is ineffective in the treatment of disease, such as in advanced cancer and in the face of new infectious diseases. Furthermore, traditional medicines are widely perceived as natural and safe, that is, not toxic. This is not necessarily true, especially when herbs are taken with prescription drugs, over-the-counter medications, or other herbs, as is very common. Cantor and Ernst 2004, Cato et al. 2008, Loya, Gonzalez Stewart, and Rivera 2009, Cohen and Ernst 2010. Regardless of why an individual uses it, traditional medicine provides an important healthcare service whether people have physical or financial access to allopathic medicine, and it is a flourishing global commercial enterprise. Engebretson 2002, Conboy et al. 2007, Evans et al. 2007. In 1990, expenditure associated with alternative therapy in the United States was estimated to be $13.7 billion. This had doubled by the year 1997, with herbal medicines growing faster than any other alternative therapy. Eisenberg et al. 1998. In Australia, Canada, and the United Kingdom, annual expenditure on traditional medicine is estimated to be $80 million, $1 billion, and $2.3 billion, respectively. These figures reflect the incorporation of herbal and other forms of traditional medicine into many healthcare systems and its inclusion in the medical training of doctors in many parts of the developed world. The total commercial value of the ethnobotanicals market cannot be ignored. For example, in 1995, the total turnover of non-prescription-bound herbal medicines in pharmacies was equal to almost 30% of the total turnover of non-prescription-bound medicines in Germany, and in the United States, the annual retail sales of herbal products was estimated to be $5.1 billion. In India, herbal medicine is a common practice, and about 960 plant species are used by the Indian herbal industry, of which 178 are of a high volume, exceeding 100 metric tons per year, Sahu 2010. In China, the total value of herbal medicine manufactured in 1995 reached 17.6 billion Chinese yuan, approximately $2.5 billion, Eisenberg et al. 1998, Hu 2001. This trend has continued, and annual revenues in Western Europe reached $5 billion in 2003 to 2004, Desmet 2005. In China, sales of herbal products totaled $14 billion in 2005, and revenue from herbal medicines in Brazil was $160 million in 2007, World Health Organization, http://www.who.int/topics/traditional_medicine/n. It is estimated that the annual worldwide market for these products approached $60 billion, Tilbert and Kaptchuk. Currently, herbs are applied to the treatment of chronic and acute conditions and various ailments and problems such as cardiovascular disease, prostate problems, depression, inflammation, and to boost the immune system, to name but a few. In China, in 2003, traditional herbal medicines played a prominent role in the strategy to contain and treat severe acute respiratory syndrome, SARS, and in Africa, a traditional herbal medicine, the Africa flower, has been used for decades to treat wasting symptoms associated with HIV, Desmet 2005, Tilbert and Kaptchuk 2008. Herbal medicines are also very common in Europe, with Germany and France leading in over-the-counter sales among European countries, and in most developed countries, one can find essential oils, herbal extracts, or herbal teas being sold in pharmacies with conventional drugs. Herbs and plants can be processed and can be taken in different ways and forms, and they include the whole herb, teas, syrup, essential oils, ointments, salves, rubs, capsules, and tablets that contain a ground or powdered form of a raw herb or its dried extract. Plants and herbs extract vary in the solvent used for extraction, temperature, and extraction time, and include alcoholic extracts, tinctures, vinegars, acetic acid extracts, hot water extract, tisanes, long-term boiled extract, usually roots or bark, decoctions, and cold infusion of plants, macerates. There is no standardization, and components of an herbal extract or a product are likely to vary significantly between batches and producers. Plants are rich in a variety of compounds. Many are secondary metabolites and include aromatic substances, most of which are phenols or their oxygen-substituted derivatives such as tannins, Hartmann 2007, Genki Kodama, Muller, and Dittman 2008. Many of these compounds have antioxidant properties, see Chapter 2 on antioxidants in herbs and spices. Ethnobotanicals are important for pharmacological research and drug development, not only when plant constituents are used directly as therapeutic agents, but also as starting materials for the synthesis of drugs or as models for pharmacologically active compounds, Lee and Veteris 2009. About 200 years ago, the first pharmacologically active pure compound, morphine, was produced from opium extracted from seeds pods of the poppy Papaver somniferum. This discovery showed that drugs from plants can be purified and administered in precise dosages regardless of the source or age of the material, Rousseau and Schachter 2003, Hartmann 2007. This approach was enhanced by the discovery of penicillin, Lee and Veteris 2009. With this continued trend, products from plants and natural sources, such as fungi and marine microorganisms, or analogues inspired by them have contributed greatly to the commercial drug preparations today. Examples include antibiotics, example, penicillin, erythromycin, the cardiac stimulant digoxin from foxglove, Digitalis purpurea, salicylic acid, a precursor of aspirin, derived from willow bark, Salix species, reserpine, an antipsychotic and antihypertensive drug from Rauwolfia species, and antimalarials such as quinine from cinchona bark and lipid-lowering agents. Example levastatin, from a fungus, Richten 2008, Schmidt et al. 2008, Lee and Veteris 2009. Also, more than 60% of cancer therapeutics on the market or in testing are based on natural products. Of 177 drugs approved worldwide for treatment of cancer, more than 70% are based on natural products or mimetics, many of which are improved with combinatorial chemistry. Cancer therapeutics from plants include paclitaxel, isolated from the Pacific yew tree, camptothecin, derived from the Chinese happy tree Camptotheca acuminata and used to prepare irinotecan and topotecan, and combratastatine, derived from the South African bush willow, Brouwer 2008. It is also estimated that about 25% of the drugs prescribed worldwide are derived from plants, and 121 such active compounds are in use, Sahu et al. 2010. Between 2005 and 2007, 13 drugs derived from natural products were approved in the United States. More than 100 natural product-based drugs are in clinical studies, Lee and Veteris 2009, and of the total 252 drugs in the World Health Organization's WHO Essential Medicine list, 11% are exclusively of plant origin, Sahu et al. 2010. Average life expectancy at birth has increased from around 41 years in the early 1950s to approaching 80 years in many developed countries. Consequently, the percentage of elderly people, 65 years and above, in our populations is increasing. The graying of our populations brings an increasing burden of chronic age-related disease and dependency. Aging is associated with a progressive decline in physiological function and an increased risk of pathological changes leading to cancer, cardiovascular disease, dementia, diabetes, osteoporosis, and so on. Lifestyle factors such as nutrition or exercise play an important role in determining the quality and duration of healthy life and in the treatment of chronic diseases, Bazzetti 2003, Benzi and Wachtel-Gaylor 2009, 2010. It is most likely that there is no one cause of aging, and different theories of aging have been suggested over the years. Genetic factors are undoubtedly important, but among all the metabolic theories of aging, the oxidative stress theory is the most generally supported theory, Harman 1992, Beckman and Ames 1998. This theory postulates that aging is caused by accumulation of irreversible, oxidation-induced damage, oxidative stress, resulting from the interaction of reactive oxygen species with the DNA, lipid, and protein components of cells. However, even if the aging process itself is found to be unrelated to oxidative stress, highly prevalent chronic age-related diseases all have increased oxidative stress, Holmes, Bernstein, and Bernstein 1992, Beckman and Ames 1998, Finkel and Holbrook 2000, Raja et al. 2009. Antioxidants in herbs may contribute at least part of their reputed therapeutic effects, Balsano and Alisai 2009, Tang and Hallowell 2010. With the growing popularity of herbal medicine, the traditional ways of identification and preparation of herbs need to be replaced with more accurate and reproducible methods, see Chapter 20, so as to ensure the quality, safety, and consistency of the product. Given the market value, potential toxicity and increasing consumer demand, particularly in the sick and elderly members of our populations, regulation of production and marketing of herbal supplements and medicines require attention. Go to 1.3, Herbal Medicines, Challenges and Regulations. WHO has recognized the important contribution of traditional medicine to provide essential care. World Health Organization, http://www.who.int/topics/traditional_medicine/n. In 1989, the U.S. Congress established the Office of Alternative Medicine within the National Institutes of Health to encourage scientific research in the field of traditional medicine, http://nccm.nih.gov, last access, November 5, 2010, and the European Scientific Cooperative on Phytotherapy, ISCOP, was founded in 1989 with the aim of advancing the scientific status and harmonization of phytomedicines at the European level, www.escop.com, last access, November 5, 2010. This led to an increase in investment in the evaluation of herbal medicines. In the United States, the National Center for Complementary and Alternative Medicine at the National Institutes of Health spent approximately $33 million on herbal medicines in the fiscal year 2005. In 2004, the National Canadian Institute committed nearly $89 million for studying a range of traditional therapies. While this scale of investment is low compared to the total research and development expenses of the pharmaceutical industry, it nevertheless reflects genuine public, industry, and governmental interest in this area. Lee and Veteras 2009. With tremendous expansion in the interest in and use of traditional medicines worldwide, two main areas of concern arise that bring major challenges. These are international diversity and national policies regarding the regulation of the production and use of herbs and other complementary medicines, and their quality, safety, and scientific evidence in relation to health claims. WHO 2005. Sahu et al. 2008. 1.3.1 International Diversity and National Policies. The diversity among countries with the long history and holistic approach of herbal medicines makes evaluating and regulating them very challenging. In addition, there are a great number of different herbs used. Legislative criteria to establish traditionally used herbal medicines as part of approved health care therapies faces several difficulties. In a survey conducted across 129 countries, who reported the following issues regarding herbal medicines: lack of research data, appropriate mechanisms for control of herbal medicines, education and training, expertise within the national health authorities and control agency, information sharing, safety monitoring, and methods to evaluate their safety and efficacy. The support needed from different countries includes information sharing on regulatory issues, workshops on herbal medicines safety monitoring, general guidelines on research and evaluation of herbal medicines, provision of databases, herbal medicine regulation workshops, and international meetings. National policies are the basis for defining the role of traditional medicines in national health care programs, ensuring that the necessary regulatory and legal mechanisms are established for promoting and maintaining good practice, assuring the authenticity, safety, and efficacy of traditional medicines and therapies, and providing equitable access to healthcare resources and their resource information. WHO 2005. Another fundamental requirement is harmonization of the market for herbal medicines for industry, health professionals, and consumers. Mahadi 2001. Herbal medicines are generally sold as food supplements, but a common regulatory framework does not exist in different countries. As a result, information on clinical indications for their use, efficacy, and safety are influenced by the traditional experience available in each place. A brief outline of the legislation in United States, Canada, and Europe is given in this section, and could be used to guide the legal aspects of the herbal medicine industry in other countries. In the United States, under the Dietary Supplement Health and Education Act, DSHEA, of 1994, any herb, botanical and natural concentrate, metabolite and constituent of extract, is classified as a dietary supplement. Dietary supplements do not need approval from the Food and Drug Administration, FDA, before they are marketed, FDA 2010. Under DSHEA, herbal medicines, which are classified as dietary supplements, are presumed safe, and the FDA does not have the authority to require them to be approved for safety and efficacy before they enter the market, which is the case for drugs. This means that the manufacturer of the herbal medicine is responsible for determining that the dietary supplements manufactured or distributed are indeed safe and that any representations or claims made about them are sustained by adequate evidence to show that they are not false or misleading. However, a dietary supplement manufacturer or distributor of a supplement with a new dietary ingredient, that is, an ingredient that was not marketed in the United States before October 1994, may be required to go through pre-market review for safety data and other information. Also, all domestic and foreign companies that manufacture package labels or hold dietary supplements must follow the FDA's Current Good Manufacturing Practice, GMP, regulations, which outline procedures for ensuring the quality of supplements intended for sale, FDA 2010, GAO 2010. Regarding contamination, the FDA has not issued any regulations addressing safe or unsafe levels of contaminants in dietary supplements but has set certain advisory levels in other foods, FDA 2010, GAO 2010. A product being sold as an herbal supplement, dietary supplement, in the United States cannot suggest on its label or in any of its packaging that it can diagnose, treat, prevent, or cure a specific disease or condition without specific approval from the FDA. A claim also cannot suggest an effect on an abnormal condition associated with a natural state or process, such as aging, FDA. In Canada, herbal remedies must comply with the Natural Health Products Regulations, Health Canada 2003. According to these regulations, all natural products require a product license before they can be sold in Canada. In order to be granted a license, detailed information on the medicinal ingredients, source, potency, non-medicinal ingredients, and recommended use needs to be furnished. Once a product has been granted a license, it will bear the license number and follow standard labeling requirements to ensure that consumers can make informed choices. A site license is also needed for those who manufacture, pack, label, and import herbal medicines. In addition, GMPs must be employed to ensure product safety and quality. This requires that appropriate standards and practices regarding the manufacture, storage, handling, and distribution of natural health products be met. The GMPs are designed to be outcome-based, ensuring safe and high-quality products, while giving the flexibility to implement quality control systems appropriate to the product line and business. Product license holders are required to monitor all adverse reactions associated with their product and report serious adverse reactions to the Canadian Department of Health. In Europe, the European Directive 2004/24/EC released in 2004 by the European Parliament and by the Council of Europe provides the guidelines for the use of herbal medicines, Calipi 2008. The directive establishes that herbal medicines released on the market need authorization by the national regulatory authorities of each European country and that these products must have a recognized level of safety and efficacy, Calipi 2008. The registration of herbal medicinal products needs sufficient evidence for the medicinal use of the product throughout a period of at least 30 years in the European Union, EU, at least 15 years within the EU, and 15 years elsewhere for products from outside the EU. With regard to the manufacturing of these products and their quality, products must fulfill the same requirements as applications for a marketing authorization. Information is based on the availability of modern science-based public monographs in the European Pharmacopoeia and their equivalents developed by the pharmaceutical industry. The standards put forward allow not only to define the quality of products but also to eliminate harmful compounds, adulteration, and contamination. Within the EU, a number of committees were set up to attempt and standardize the information and guidelines related to herbal medicines. A variety of materials has been produced, such as monographs on herbs and preparations, guidelines on good agricultural and collection practice for starting materials of herbal origin, and guidelines on the standardization of applications and setting up pragmatic approaches for identification and quantitative determination of herbal preparations and their complex compositions, Routledge 2008. Vlietink, Peters, and Apers 2009. 1.3.2, Quality, Safety, and Scientific Evidence. Herbal medicine has been commonly used over the years for treatment and prevention of diseases and health promotion as well as for enhancement of the span and quality of life. However, there is a lack of a systematic approach to assess their safety and effectiveness. The holistic approach to health care makes herbal medicine very attractive to many people, but it also makes scientific evaluation very challenging because so many factors must be taken into account. Herbal medicines are in widespread use, and although many believe herbal medicines are safe, they are often used in combination and are drawn from plant sources with their own variability in species, growing conditions, and biologically active constituents. Herbal extracts may be contaminated, adulterated, and may contain toxic compounds. The quality control of herbal medicines has a direct impact on their safety and efficacy. Ernst, Schmidt, and Weider 2005, Rybnicki et al. 2008. But, there is little data on the composition and quality of most herbal medicines, not only due to lack of adequate policies or government requirements but also due to a lack of adequate or accepted research methodology for evaluating traditional medicines. Who 2001, Cantor 2009. In addition, there is very little research on whole herbal mixtures because the drug approval process does not accommodate undifferentiated mixtures of natural chemicals. To isolate each active ingredient from each herb would be immensely time-consuming at a high cost, making it not cost-effective for manufacturers. Richter 2003. Another problem is that despite the popularity of botanical dietary and herbal supplements, some herbal products on the market are likely to be of low quality and suspect efficacy, even if the herb has been shown to have an effect in controlled studies using high-quality product. There is a belief that herbs, as natural products, are inherently safe without side effects and that efficacy can be obtained over a wide range of doses. Although herbs may well have undesirable side effects, there are no set doses, and herb-drug or herb-herb interactions are possible. A major hypothetical advantage of botanicals over conventional single-component drugs is the presence of multiple active compounds that together can provide a potentiating effect that may not be achievable by any single compound. This advantage presents a unique challenge for the separation and identification of active constituents. Compounds that are identified by activity-guided fractionation must be tested in appropriate animal models to confirm in vivo activity. Ideally, the composition of the total botanical extract must be standardized and free of any potential hazards, and plants should be grown specifically for the production of botanical extracts under controlled conditions and originate from a characterized and uniform genetic source with a taxonomic record of the genus, species, and cultivar or other additional identifiers. Records should be maintained for the source of the seed, locations and conditions of cultivation, and exposure to possible chemical treatments such as pesticides. Because the environment can significantly affect phytochemical profiles and the efficacy of the botanical end product, botanical extracts can vary from year to year and may be significantly affected by temperature, drought, or flood as well as by geographic location. Therefore, biochemical profiling must be used to ensure that a consistent material is used to produce a botanical. The concentration step can also be challenging, and the process to concentrate active compounds to a sufficient level can negatively affect their solubility and bioavailability. Therefore, improving efficacy by increasing concentration can be counterproductive, and the use of solubilizers and bioenhancers needs to be considered just as for drugs, Rybnicki et al. 2008. However, there are major challenges to achieving this. Although in theory botanicals should be well characterized and herbal supplements should be produced to the same quality standards as drugs, the situation in practice is very different from that of a pure drug. Herbs contain multiple compounds, many of which may not be identified and often there is no identifier component, and chemical fingerprinting is in its early stages and is lacking for virtually all herbs, see Chapter 20. This makes standardization of botanicals difficult, although some can be produced to contain a standardized amount of a key component or class of components, such as ginsenosides for ginseng products or anthocyanins for bilberry products, see Chapter 4 on bilberry and Chapter 8 on ginseng in this volume. However, even when such key compounds have been identified and a standard content is agreed or suggested, there is no guarantee that individual commercial products will contain this. Another interesting point to consider is that herbal materials for commercial products are collected from wild plant populations and cultivated medicinal plants. The expanding herbal product market could drive overharvesting of plants and threaten biodiversity. Poorly managed collection and cultivation practices could lead to the extinction of endangered plant species and the destruction of natural resources. It has been suggested that 15,000 of 50,000 to 70,000 medicinal plant species are threatened with extinction, Brower 2008. The efforts of the Botanic Gardens Conservation International are central to the preservation of both plant populations and knowledge on how to prepare and use herbs for medicinal purposes, Brower 2008, Lee and Veteras 2009.